Now that we're familiar with the basic anatomy of a human skeletal muscle fiber, and we've also seen what happens at the neuromuscular junction, we can see what happens within the muscle fiber once it's stimulated. So let's go back to inside our sarcomere, where we can see a thick filament made up of our myosin molecules, our thin filaments made up of actin. They're wound around by this tropomyosin. The tropomyosin, as we know, is held in place by the troponin molecule. We know that the myosin heads will want to interact with the actin, but of course during the resting state, the muscle was not active, and so the troponin is holding the tropomyosin gate closed so that the active sites on the actin are covered, and therefore these myosin heads cannot interact with them. So here once again our purple tropomyosin gate is covering the active sites and is being held in place by the troponin. So what happens when we have an action potential? Well, we've seen the events of the neuromuscular junction. We have our action potential propagate first down our motor neuron. It releases acetylcholine at the neuromuscular junction here. The acetylcholine diffuses across the synaptic cleft, opens sodium channels on the motor end plate of the sarcolemma, causing the rapid influx of sodium, and this is what's going to initiate the action potential within the muscle fiber. So that action potential will be propagated along the sarcolemma. And as it propagates along the sarcolemma, that action potential will be brought deep into the interior of the cell by the transverse tubules, also known as the T-tubules, which are basically indentions of the sarcolemma, or that excitable membrane, into the cell. And we know that the transverse tubules have a nice arrangement with our sarcoplasmic reticulum so that they are making these junctions called triads around a transverse tubule. So we have our terminal cisternae here, which we know are full of calcium. Calcium is the key, as we know. So what does the action potential do once it's propagated across the membrane and then down into the cell? Well, it's going to cause the release of calcium from the terminal cisternae. And as we've noted, the terminal cisternae are located strategically over our zones of overlap in our sarcomere, which is underneath here. So if we zoom in a little bit and look at the events within the sarcomere between the thick and thin filaments, once that calcium is released from the sarcoplasmic reticulum, what's going to happen is the calcium will come and bind with the troponin molecule. And that's like unlocking it. So here we have our tropomyosin molecule that is covering our actin filaments right here so that our myosin heads cannot interact with it. The troponin is like the lock that's holding the gate in place, but as soon as calcium comes in and diffuses out of the sarcoplasmic reticulum into the sarcoplasm, then what happens is calcium can now bind with this thing. And it acts with a key, as a key. And the troponin is then like a lock. And so it changes its shape. And now the tropomyosin can rotate. And look at what happens here. The tropomyosin has now rotated into a different position. And now we can see the active sites on the globular actin here. So now we see our active sites revealed. And now we see that our myosin heads can interact with it. So what they do, they form cross bridges. And they will form a cross bridge and they will grab onto this thing. And then they will pivot, just like a ratchet, and pivot towards the M line. And that will pull these actin filaments towards the M line. In this case, it's pulling in this direction. So it's going to pull the actin filament in this direction. And then it's going to release it. And it's going to start the cycle all over again. And as long as we have calcium bound to troponin, and the active sites are revealed, then these myosin heads will continue this cycle of cross-bridging, pivoting, releasing, and cocking itself back out for use again. Now, here's another important thing that we need to notice. Adenosine triphosphate is our high-energy molecule, and this is very important. Without ATP, without calcium, we cannot make this cross-bridge cycle, so we cannot have this sliding of the actin over the myosin if there's no calcium and if there's no ATP, because obviously we need the calcium to act as the key to keep the troponin unlocked, as it were, and the tropomyosin gate rolled open so that the active sites are revealed. So calcium is key for that. ATP is the energy molecule that allows all this to happen. So in order to get into this cocked and ready position, the myosin heads have to break ATP. So they use it as an enzyme, or they actually are the enzyme. The myosin heads are the enzyme, and they use ATP to cock into the ready position. And when they do this, then they are ready to grab onto the active sites when they are revealed. So when it breaks the ATP, if you remember, ATP is adenosine triphosphate. So it's a nucleotide with 3 phosphates attached. And the third phosphate is the high energy phosphate that when you break that bond yields the energy. So what happens is these myosin heads will take that ATP and they will break it. And what they're left with is adenosine diphosphate, that's adenosine with 2 phosphate groups still attached, and this lone phosphate group that they broke off for the energy. Now, notice that they hold on to this. And so they're still holding on to it after they've ratcheted themselves out into the ready position. And so what happens is during the resting state, they're already ready to go. So before the muscle's ever stimulated, the myosin heads are in the cocked and ready position because in the previous contraction cycle, they bound ATP and they broke it and then they ratcheted back out or cocked themselves into the ready position. But once the tropomyosin covers the active sites, well, then they're not going to interact with it. But they're ready to go for the next contraction cycle. So when the muscle is stimulated again and the active sites on actin are revealed, then they can interact with it. Notice they still have the adenosine diphosphate and phosphate bound to it. Once they have made the cross bridge with the actin, now they have to pivot. And to do that, they have to drop the ADP and phosphate. So when they pivot, they drop the ADP and phosphate. But now they're actually stuck to the active sites. And they can't let go until ATP binds with them. So adenosine triphosphate must bind with the myosin heads in order to get them to let go of the active sites. Now, once the ATP is bound, then the myosin head can let go, but notice it's still in the pivoted forward position. So therefore, the myosin heads will break that ATP to ratchet back out into the ready and cocked position. And so that is the cycle of cross-bridging and how the myosin heads will interact with the active sites on the actin filaments. And so long as the active sites are uncovered, so long as we have calcium and adenosine triphosphate present in the muscle, then this cycle of cross-bridging will continue. And as long as the cycle of cross-bridging continues, then the muscle fiber will shorten because we have these myosin heads basically pulling on these actin filaments. And of course, that is like bringing this Z lines closer to the M line. So if we were to say that this room was a sarcomere and my body is the M line and my arms are like myosin filaments and my hands are the heads, and I'm grabbing onto actin that's coming from the wall and I'm doing this and doing this over and over and over and over again, and basically picking up the actin filament, ratcheting it forward each time, that's going to draw the walls or the Z lines closer to the M line. And that's what's going to cause the shortening of the sarcomere. So that's exactly what we see here. We see that the interaction between the myosin thick filaments and the actin filaments, and the fact that the myosin is pulling the actin filaments towards the M line, that's bringing the Z lines closer to the M line, and that is shortening the sarcomere. And that is how a muscle contracts.